So this next unit is on bioenergetics, which involves photosynthesis and cellular respiration. So if we think to the organisms that live in the environment, the first thing we know is energy is essential to life. If we don't have energy, we can't have life taking place. And if you think to our ecology unit that we did, we actually had two different types of organisms. We had our autotrophs and that could make their own food, and heterotrophs. So autotrophs consist of things like plants, whereas heterotrophs are things like animals and fungi. Autotrophs use light energy to produce food, whereas heterotrophs cannot use the sun's energy directly. That's because autotrophs use the process of photosynthesis, whereas heterotrophs obtain energy from the food that they eat. So we're going to deal first with the autotrophs and the processes that they undergo to produce the food from light energy. And that involves a specialized cell organelle called the chloroplast. Chloroplasts are small green organelles in plant cells and also in some protist cells. This is the actual site of photosynthesis, and you will actually see throughout my notes I abbreviate photosynthesis PHS, and that's just to help reduce some of the writing. If you wish to use that, you may. Now, if we opened up the chloroplast, we would actually see that it's not just a simple, sac-like organelle. There's actually additional things inside of it. Inside of it, there are photosynthetic membranes that we call the thylakoids. And inside of the thylakoids are actually the clusters of pigments that we have that allow photosynthesis to take place. Now, these thylakoids are arranged in stacks that we call the grana, or grana, and the space in between these stacks are called the stroma. So here's a picture of a chloroplast, and you can see the outer layer of the chloroplast is actually a double membrane, and then inside we have these stacks of circular, flat circular, almost disc-like thylakoids, and that is called a granum, and then the space around them are the stroma. So over in your handouts packet, we're going to look at the first one here that deals with the chloroplast. So let me get my projector ready here. And so it says, in plants, photosynthesis takes place in chloroplasts. Inside chloroplasts are sac-like membranes called thylakoids. These thylakoids are arranged in stacks. A stack of thylakoids is called a granum, so multiples are called a grana. The region outside of the thylakoids, but inside the chloroplast, is called the stroma. So we need to label the thylakoid, the stroma, and the granum. So we have a line coming to this single stack here, the single sac, and that is the thylakoid. The stack of thylakoids is called the grana. And then the last one is pointing to the space in between, and that is the stroma. And then underneath, we have some questions. And these are a little bit of advance for where we are right now, but where are the photosystems or light collecting units of photosynthesis found? Well, if we're collecting light, we're using a pigment, as we will learn, so that is in the thylakoid membranes. In what part of the chloroplast does the Calvin cycle take place? And again, we haven't learned about that, so that's okay, but that takes place in the stroma, as we will learn. And in what part of the chloroplast do the light-dependent reactions? Well, if these are light-collecting units in the thylakoids, that's also where those light-dependent reactions take place, is in the thylakoids. So let's talk about this interaction between light and pigments here for a minute, because that is really the key to photosynthesis. The main pigment of photosynthesis is a pigment called chlorophyll. And the pigment itself is green. It's actually a protein, and that protein is able to absorb sunlight energy. What you have to remember is that sunlight is white, and it's a mix of all the different colors: red, orange, yellow, green, blue, indigo, and violet. Now, you're going to actually be doing an activity that takes a look at this, And what you will find is that photosynthesis relies on red and blue light the most. It actually absorbs the light in those regions of the spectrum better. So if you were to put a plant under red light or blue light, it would actually grow really, really well. It relies the least on the green. And so what this pigment does is it actually reflects back the green color to us rather than absorbing it. So that's why we see plants as being green. If you were to try to grow a plant under green light, it actually would not grow very well. Now you can see that here on this chart. Now, I said chlorophyll. Chlorophyll is actually a combination of a couple of different chlorophylls, and you can see here chlorophyll A and chlorophyll B. And you can see here we've got these big peaks. This one is in the blue range, and then chlorophyll A has a big peak in the red range, and then chlorophyll A also has another one down in the violet range. Notice where both of them are low, though, is in the green range. Now, if we think of a plant where we have most of the green color is on the leaves. Now, the structure of a leaf is actually optimized. It's actually designed to absorb light and undergo photosynthesis. If you think about like a maple leaf or an oak leaf, they are large, they are flat, and they are broad, and that is designed to absorb a lot of light while it is available. That's all they are doing. They are literally photosynthesis factories. You can see some of this here. You can see the leaf is very broad and flat, and we have all of these different things with it, and we're going to kind of go through it. Notice that these cells up at the top here are very tightly packed, whereas these down the bottom have some air space in them. All of these dark green dots that you see, those are all chloroplasts, and you can see that they are packed into these cells on the leaf. So that top layer of the leaf, where the cells are very tightly packed, is called the palisade mesophyll. So it's located on the top side underneath something called the epidermis. Basically, that's like the outer skin of the leaf. These cells are very tall and column-like, and they are tightly packed. Most of our photosynthesis takes place here because if you think about it, top side of the leaf tends to get most of the sunlight. Underneath is a layer called the spongy mesophyll. These are the ones that are loosely packed. If you think of a sponge, it's got lots of air spaces. And the surface of these cells are moist, and that allows the gas exchange. And you will see that gases are needed in order for photosynthesis to take place. So we need these air spaces and the ability to exchange gases in order to do that. How do we get gas into the leaf? Using something called the stoma. The plural of that is stomata. These are almost like pores, like we have pores in our skin on the underside of our leaf. And like I said, these allow the gas exchange, the carbon dioxide and the oxygen. So carbon dioxide actually comes into the leaf, oxygen comes out. It also allows transpiration, which is the loss of water through the leaves. Plants actually pull water up through their roots, and some of that water is lost through the stoma. The stoma actually open and close by specialized cells called guard cells. And you will see here these guard cells. You can see here we've got the stoma that is open, and the guard cells are in one position, and then when they close up, no more air or gas exchange can take place. So now that we understand the principles of the leaf and a little bit about these chloroplasts, let's actually get into the process of photosynthesis. If we look at kind of an overview of photosynthesis, we've got the chloroplast here. The idea is we have light energy coming in, and that allows us to take carbon dioxide and water and change it into glucose, which is a sugar that is needed for life, and oxygen. So with photosynthesis, we actually have two sets of reactions. The first grouping of them is called the light reactions, sometimes called the photosystem reactions because they deal with light. And then we have the Calvin cycle, which are sometimes called the dark reactions. Now don't let this confuse you. The Calvin cycle can take place in the presence of light. It just doesn't require light in order to take place. So let's look at these light reactions. They're going to happen in the thylakoid membranes in the chloroplast because these are the places that capture the light, and they require light to run. Light reactions use water, they produce oxygen, and it generates two compounds that we're going to use in the second step. The first one is ATP, and the second one is NADPH. And those are actually going to allow us to generate the glucose that we want. Now, NADPH is a group of compounds that we call electron acceptors. These are able to hold on to high energy electrons. And what it does is it allows them to capture the energy of electrons, transport it, and use it somewhere else. These guys are reusable. In photosynthesis, the electron acceptor is called NADP+. And once it's accepted a high energy electron, we now call it NADPH. So the light reactions create one molecule of ATP that we use later, and this is actually formed through the process of chemiosmosis that we will talk about later in the unit. So if we were to summarize the light reactions in terms of what we need or we need to put in, and we need to bring in water, we need to have that electron acceptor of NADP+, we need light, and we need ADP and a phosphate. So what do we get out of it? We get oxygen, NADPH, which we will use later, and ATP, which we will use later. So let's take a look now at the handout in your packet that looks like this, Photosystems 1 and 2. So Photosystems 1 and 2, now these are basically the light reactions that we just talked about. So we are going to color our diagram according to the directions here. So color the two places where light energy enters yellow. Okay, so light energy is represented by these squiggly arrows here, so we are going to color them yellow. Okay, and this one too. Color the hydrogen ions red. Okay, so this is a hydrogen ion, this is a hydrogen ion, this is a hydrogen ion, and this is a hydrogen ion. And then we've got another one here, and another one here. I think that's all of them. Yep. Color the electrons green. So we have green electrons here. And notice that they are moving across the thylakoid membrane. And what's happening is they're capturing the energy from the light as it comes in, and then they transport that to make ATP and NADPH. And then it says color the thylakoid membrane blue. Okay, this is the thylakoid membrane, so you would want to color all of this blue, which I'm just going to kind of roughly do here. This is the thylakoid membrane, so right there. All right, so the questions. Where does light energy enter the system? Well, if you look back, you see those arrows are coming in at photosystem 1 and photosystem 2. So you'll see those arrows are coming in where they have those labeled. What uses energy from the high energy electrons to transport hydrogen across the thylakoid membrane? That is the electron transport chain. You can see on the diagram here that we have this hydrogen moving across, and then we have the light energy coming in at photosystem 1 and photosystem 2. Okay? So those are the light reactions. The second part that takes place as part of photosynthesis is the Calvin cycle. The Calvin cycle, like I said, is also called the dark reactions. This is the process of what we call carbon fixation. And carbon fixation is the incorporation of carbon dioxide into organic compounds. It occurs in the stroma, so not in the thylakoids. It occurs in the spaces, but we're still inside the chloroplast. And it consists of the creation of PGAL. Now, PGAL is important because it can be used to make things like glucose, fructose, starch, and cellulose. But in terms of what we're looking at in photosynthesis, we're looking at glucose, which is its primary use. It can be used to make these other things, but primarily, and what we think of with photosynthesis, is that glucose. So with the Calvin cycle, again, what do we need? We need ATP, we need NADPH, and we need carbon dioxide. What do we get? We get that ADP plus a P, so we use the energy from ATP, and again, we'll talk about energy and ATP here in a few minutes. NADP plus and an H plus. Both of these get recycled back to the light reactions. We also get glucose, which is used for our cellular respiration or building cell materials. Okay, sorry about that. So in any case, the Calvin cycle, we get all of this out. So the glucose is used for our cellular respiration, which is the second half of what we're looking at, or for building our cell materials. So we're going to move over into our handouts packet again and work with the Calvin cycle. ATP and NADPH are both produced by the light-dependent reactions of photosynthesis. The Calvin cycle uses energy, and ATP and NADPH to produce high-energy sugars. So we're going to circle the places where ATP and NADPH are used. Then we're going to draw an X over the 6-carbon high-energy sugar produced by the Calvin cycle. So if we start at the top, you can see here that we've got 6 carbons of carbon dioxide coming in, and we're going to follow this around clockwise. And notice here we have ATP. ATP changes to ADP, so we're using that there. We also have NADPH here, and we're changing that back to NADP+. Both of those, remember, are going to go back to the light reactions and get recharged. Then we come around here and we actually have 6 more ATP that get used. So those are all the places in the Calvin cycle where we get ATP and NADPH used. The second thing is we need to draw an X over the 6-carbon high-energy sugar. So each of these little C's with a circle represent a carbon. So here we have 1. We go over here, we have 3. So we have 3, 3, and then 1, 2, 3, 4, 5. We have 5 on the last part of the circle. But if we look down here at the bottom, we have 2 3 carbons, and then we have a 6 carbon underneath it. So that's what we need to draw the X over. So how many molecules of carbon dioxide are used to produce one 6-carbon sugar molecule? Well, we need 6 of them. If you look at the top, it says 6C, and that's 6 CO2s. What is formed as a result of the Calvin cycle? A 6-carbon sugar is formed. So if we take an overall kind of a little bit more detailed look at the process in this diagram here, okay, this again is our chloroplast. So we have light energy coming in, and remember, this is the thylakoid membranes. These are our granum here. We also have to bring in water, and we get oxygen out of it. This is where the light reactions take place. So we not only get oxygen released, but then we cycle over to the other side by bringing in carbon dioxide and funneling out glucose through the Calvin cycle. Now again, there are things that are shuttled back and forth between both the light reactions and the Calvin cycle. So the light reactions make ATP. They also make NADPH that get used in the Calvin cycle. Once they are used in the Calvin cycle, they get shuttled back, so NADP plus and H plus go back to the light reactions, as does the ADP plus the P. So this is kind of an overview. Now, this stuff in the middle here, the ATP and the NADPH, we usually leave that off when we're talking about the overall photosynthesis equation, simply because they are cycled. Now, keep in mind that photosynthesis has to take place in the presence of both chlorophyll and light. I can't just put these things together and have it magically happen. We need to have the chlorophyll, and we need to have that light energy coming in. So here is the reaction. We need 6 carbon dioxides and 6 waters, and that's going to give us glucose, C6H12O6. That's a formula that you should recognize, and 6 oxygens. So we get the carbon dioxide from the air, and the water comes from the soil for the plant. The glucose gets stored in the leaves, and remember, that's how the energy gets transferred to the through the food chains and the food webs, and then the oxygen is released into the air, and the heterotrophs use that. All right, so let's take a look here at the photosynthesis overview that's in your handout packet. So it kind of looks like what we just saw. Okay, so we're going to start on the left here, and the squiggly arrow, remember, represents light energy coming in, combining with water in the light dependent reactions, and that kicks out down the bottom here oxygen. We have ATP coming over and ADPH coming over here to the Calvin cycle, where we have carbon dioxide coming in, and then it sends over back to the light reactions the NADP plus and the ADP plus P. And the Calvin cycle kicks out the sugars. Now remember, the sugar that we're worried about most is our glucose. Okay. So with the questions at the bottom, finish the equation for photosynthesis. So you have carbon dioxide plus water, and notice you've got the light above the arrow. That means in the presence of light. So after that, you just need to add glucose and oxygen. That's what comes out at the bottom of the chloroplast there. Which of the following is also called the light independent? So in other words, it works independently without light in photosynthesis. That would be the carbon cycle. All right, the last one that we're going to do for this part of photosynthesis is our flow chart here. Okay, so if you look at number 1, we have water. Water plus light go into the light-dependent reactions. And then the light-dependent reactions kick off the oxygen. You see the O2 there. And then it also kicks off NADPH for number 2 and ATP for number 3. Those go into the Calvin cycle along with CO2, which is number 4. And the Calvin cycle then kicks off sugars and then cycles back to the light-dependent reactions ADP plus P and NADP plus. So this brings to an end our first section of notes here on photosynthesis, and then in the next one we will actually talk about chemical energy and some factors affecting photosynthesis, and then get into cellular respiration.